Okay, so now what we want to talk about is what's called transcription. Transcription, just simply put into a couple words, is reading DNA to make its complementary RNA sequence. Because essentially after that point, we're going to take that RNA and we're going to make proteins out of it, because we know, learning about proteins, how integral and important they are to sustaining biological life. They do everything. We've already talked about that. We know that. Okay, so I'm going to read through a couple of these key points and circle a few words that you're going to need to know, essentially your vocabulary words. So the sequence of RNA bases together with the structure of the RNA molecule determines the RNA function. Essentially, we're reading a gene in DNA or instructions for a protein. Of course, it's going to determine the function because the function is determined by its genetic coding in the DNA. Our key players in this process are mRNA, messenger RNA, which carries the information from the nucleus to the ribosome. DNA never, ever, 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 never leaves the nucleus in this process. It's too important. It must stay there. It's only copied and then that information is then transported out. You're never going to allow DNA to leave that nucleus. Some other key players that are going to be important in this process are rRNA, our pirate RNA, so to speak, but this is going to be our ribosomal RNA. This is a ribosome that is reading the message that's going to be then turned into a protein. And then finally, we're going to talk about transfer RNA. These molecules carry the amino acids, which then build together to make your protein or your peptide sequence. Okay, this is your protein. So these three are what we're going to focus on for transcription. Again, transcription is just taking your DNA sequence, modifying it a tiny bit to get your RNA, which specifically is your mRNA or your messenger RNA. Okay, so there are a few more things here on this list, but I'm going to address them specifically in a picture because pictures are a lot better way to look at this process. Okay, so here is, let me zoom in just a tiny bit. Here's transcription. So, inside the nucleus, we have our DNA molecule. Our DNA molecule is going to be opened up by a few different enzymes, and it's going to be read or transcribed into RNA. The enzyme that does this is an enzyme, is RNA polymerase. RNA polymerase, just like DNA polymerase, is going to add the complementary bases from DNA to RNA. Now, I want to bring you back to talking about the bases of DNA and RNA. Okay? When we go from DNA to RNA, our four bases for DNA are A, T, C, and G. In RNA, we now have A, U, C, and G. Okay? For going between the two, I like to make this little tree here, kind of like a little flow chart that tells you what the DNA base is and what you're now going to have in RNA. So in DNA, we have A, T, C, and G. But as RNA polymerase reads this strand, A is now going to give you, or adenine is now going to give you the base uracil, because protein code has adenine, uracil, cytosine, and guanine. No more thymine. Thymine is specifically for DNA. So as RNA polymerase, this molecule right here, reading the DNA strand, T is still going to give us A, and C and G are simplistic. C gives you G, G gives you C, okay? Put this on your notes. This is going to be helpful until you learn how to specifically go through transcription. All right, so in terms of transcription, RNA polymerase reads it. You get this rough draft of an RNA molecule. A few things have to happen to it. We have to edit it because there may be a few components or gene sequences built in there that are not specifically what we want in terms of the gene we're looking for. So those things are called this: introns and exons. Exons are spliced or put together, spliced, to exit. This is what leaves the nucleus. This is the final product or our proofread piece of mRNA. Introns, different color here because it's matching my nucleus, stay in the nucleus. Okay? We don't want the introns. The introns are not the components that we want. The exons are what are going to exit the nucleus. So once we have the piece of RNA, the messenger RNA, there's a couple other things that are going to counteract possibly degrading once it gets to the cytoplasm, because the cytoplasm might not have the perfect chemical components for every molecule that is there, so sometimes we need some protecting. The protecting could be a vesicle bubble. Other protecting could be in terms of this. There's going to be a cap put on it and a poly A tail. These are going to help the molecule from degrading, okay? It's gonna give a starting point and a stopping point, and it's gonna help it make its way to the ribosomal RNA. And that's what this is. This is our ribosome here. The ribosome is going to read through this message, and it's going to help build our protein. What does the building, or assists in the building, are these structures here. These are your transfer RNA. Your tRNA. And what they have attached to the top of it are your amino acids, and you can see them kind of floating around here in the cytoplasm. Once a transfer RNA is initiated or helped by this messenger RNA, it's going to attach to it in a specific way, which we'll talk about here in a couple minutes, and it's going to drop off those amino acids until you have the polypeptide or the protein sequence that you need. Okay, so that in a nutshell is transcription. Specifically, transcription is just the making of this mature or finished mRNA before we get to the ribosome. The ribosome reading it is translation, which we're going to get into in just a second.